Hi everyone! We will continue on the third part of the lecture. In this video, we will learn about the mechanism of DNA transfer and see how DNA is being uptaken by gram-positive and gram-negative bacteria. We will also touch briefly on the process called homologous recombination. For transformation to happen, the very first step involves the uptake of DNA from outside the cell. The uptake of DNA might vary from one species to another. DNA must first bind to the cell surface, and depending on the cell, some will have a pillar structure while others might have secretion system. Here are images showing DNA binding and uptake system on the gram-positive, which is bacillus, that have pillar-like structure, while Neisseria gonorrhea is the gram-negative that have secretion system. All protein complex here must be able to take up DNA and move it through cell wall. Next, the new DNA may or may not be cleaved, but eventually only one DNA strand will reach cytoplasm. The degradation could occur at the external surface or somewhere in the cell wall before DNA reaches cytoplasm. However, the mechanism is still debatable and being studied. Let's take a look at the mechanism. Bacteria differ in their ability to uptake different forms of DNA. For instance, gram-positive bacteria like Streptococcus uptake and move single-stranded DNA across cells due to the presence of thick peptidoglycan. Gram-negative like Haemophilus influenza and Neisseria gonorrhea, on the other hand, may uptake double-stranded DNA, but only one strand involved in recombination with the host DNA. Now, we'll get into greater details for transformation process in gram-positive bacteria. We will use Streptococcus pneumoniae as the example. If you remember, I mentioned previously about competent state that occur during limited time only, when nutrient is scarce and cell density is high. Well, for S. pneumoniae, it becomes competent during exponential phase, when population is very dense, accumulating at 10 to the power of 7 to 10 to the power of 8 cells per mil. So, this competent cell starts to secrete a small protein called competent factor that later stimulate production of 8 to 10 new proteins chaperone needed for transformation. Then, the competent cell can bind to any random double-stranded DNA fragment that is moderately large in size. Sometimes donor fragments are competing with each other to bind to the cell. This is because DNA uptake requires energy. DNA is then cleaved to smaller fragments sizing around 5 to 15 kb. Next, one of those two strands will be hydrolyzed by an envelope-associated exonuclease during uptake, and the other strand will be associated with small protein and move through the plasma membrane. The single-stranded DNA that is already in the cell can later align with a homologous region of the genome and be integrated into the host chromosome. The cell is now a newly transformed cell. Okay, we have seen the transformation in S. pneumoniae, which is the gram-positive bacteria. How about gram-negative? Haemophilus influenzae, which is a gram-negative bacteria, exhibit different DNA uptake process from S. pneumoniae. H. influenza does not produce a competent factor to stimulate the development of competent. The successful DNA uptake process is basically determined by these two following factors. First, specific sequence must present on the DNA fragment and be identified by recognition site on the cell. 2. Small membranous structure must be formed This structure is called transformosome, which protrude outside the cell. Haemophilus influenzae takes up DNA from only closely related species, so it's basically different from S. pneumoniae where the binding process is random. The specificity of Haemophilus transformation is due to DAS, or species-specific DNA uptake sequences. DAS for Haemophilus influenzae is a special 11 base pair sequence, so if you can see in the slide, and this sequence is repeated more than 1,400 times in the genome. Basically, DNA fragment must have this sequence to be bound by a competent cell of Haemophilus influenzae for transformation to happen. Once the specific DNA fragment, usually size around 30 to 50 kb, has bound to membrane receptor, the double-stranded DNA complex with protein is engulfed by membrane vesicle. The vesicle is what we call transformosome. The transforming DNA is taken up into transformosome where it is then internalized into the cell. One of two strands of DNA is degraded in the vesicle while the other strand remain. This remaining strand later may recombine with the host chromosome, forming a newly transformed cell. The transformation won't complete if the foreign DNA is not being integrated into the recipient cell genome. The integration of DNA into the host chromosome will occur through a process called homologous recombination. Homologous recombination rely on extensive sequence similarities. One single-stranded DNA, or what we call ssDNA, that represented by the orange color here, entered the cytoplasm. It is bound by a single strand binding protein that protect the single-stranded DNA from immediate degradation. Then, another protein called DPRA or DNA processing A will displace SSB. DPRA also facilitate the loading of RAG-A onto the nucleoprotein filament. RAG-A will promote the alignment of homologous sequence and catalyze strand invasion into the recipient chromosome. The recipient chromosome is represented by blue color here due to sequence complementarity. After strand exchange has occurred, a heteroduplex of transforming DNA and recipient DNA is now established. When replication and the next cycle of cell division occur, two strands of the heteroduplex may part, yielding a transformed and a wild-type cell.